Yang Su, Manager, New Technology and Applications, Microfluidics. Yang, are you here? Yes? Okay, perfect. Dr. Su is a distinguished and highly sought-after speaker on a wide variety of applications ranging from lipid nanoparticles, cell disruption, nanoemulsions, and more. Dr. Su has been with Microfluidics International Corporation since 2012. As a manager of new technology and applications, Dr. Su focuses on the continued development and promotion of microfluidics, cutting-edge microfluidizer technology, to deliver the desired outcome for each customer's unique application. And the title of the presentation is a versatile solvent-free scalable technology for producing RNA delivery platform.
Thank you for the introduction. And good to see everyone here, and thanks for joining me this morning. As my title suggested, I'd like to introduce you a solvent-free and scalable technology and explain a little bit more on how it can help producing various RNA delivery platforms. So here's the agenda of my today's presentation. I'll start with a very brief introduction on RNA-based therapeutics and its delivery. We've all heard the a great keynote speaking this morning, so I'll keep it short. Then discuss a little bit more about some of the considerations and requirements or challenges during the development and manufacturing of those platforms, and introduce you Microfluidics as a company and the Microfluidizer technology. I'll use a bit more time to discuss more lipid-based nanoparticles with RNA delivery, and my presentation by showing you our diverse product portfolio. So conventional treatment, we all know that targets proteins rather than the underlying causes, so they generally induce transient therapeutic effects. It is also very well known that only a very, very small portion of human genome are druggable targets, and even when you factor in or consider antibodies. So nucleic acid-based therapeutics, on the other hand, targets disease from the genetic basis, so can achieve long-term or even curable results. So among them, RNA therapeutics definitely has great potentials with some unique benefits. First, RNA can theoretically target rows and druggable targets by choosing the correct sequence. The RNA therapy also has no significant genotoxic effects compared to DNA therapy. As we heard this morning, that with the key discoveries and advancement in RNA technology, as shown down here, RNA-based drugs are being developed with less time and money, and you can also see that here, and also this morning earlier, that more and more products has been approved in recent years. However, clinical translation of RNA-based therapeutics depends on delivery technologies. We just heard a great presentation about lipid nanoparticles. But delivering RNA has remained as a major challenge due to some factors. That first, the RNA is highly unstable, and it can potentially trigger immunological responses. Additionally, naked RNA are not capable of crossing cell membranes to achieve efficient intracellular delivery. So to overcome those limitations, various delivery strategies have been extensively studied. I've showed a few here. So they can generally be grouped into either non-viral-based nanoparticle platforms or viral-based platforms. So I have a few examples here showed, as a part of the non-viral-based nanoparticles, obviously there's LNPs and several other lipid-based nanoparticles, or polymer nanoparticles and a few other systems. On the other hand, viral delivery platform utilizing several different kinds of viruses. Although I think many people are interested in LNPs because LNP certainly has showed its great potential during the past few years. We also heard from the previous speaker that it has its own limitations, especially on the stability side. So that's my focus later on with my presentation is talk about this and also showing you the alternatives with a slightly different approach and similar lipid-based particles. However, regardless what platform you choose to work with, there are some key factors and considerations you need to think about during the development and as well as commercial manufacturing. So for non-viral platforms for those nanoparticles, I think first and foremost are physical chemical properties. There are many factors. One of them definitely, I think, is particle size and distribution, because the particle size can significantly affect the stability of the particles as well as their both in vitro and in vivo performance. Once you have come to a good formulation with the desired particle size in the development stage, you want the process to be scaled up easily and directly with batch consistency. Also, many methods currently are used for producing these particles involve using large amount of organic solvent. So using the solvent not only poses additional burdens to the downstream processes because you have to remove the residual chemicals, but it is also start facing a lot more pressures due to increased health and safety concerns. So a process or technology can produce those particles without using the solvent can provide huge benefits. Another consideration is if the process or a processing equipment can help improve the entire process or make the adjacent process easier. For example, in vivo delivery of the product requires sterilization, which is commonly done by terminal sterilizations through sterile filtration. So the filter membrane used in the filtration has pore size around 0.22 microns or 220 nanometers. Which implies that to achieve efficient filtration process, the majority of the particles in the product should be less than that size. And last but not least, for especially pharmaceutical product manufacturing, the entire process needs to comply with regulatory requirements, for example, CGMP. So our microfluidics technology can help address all these challenges and requirements from production to from development to production. Before I introduce you or explain how the technology works, I want to quickly just tell you a little bit more about the company. So Microfluidics is where I come from. Along with a few other companies, is part of the Material Processing Technologies or MPT platform under under the Health and Science segment of IDEX. IDEX is our parent company. So Microfluidics itself was founded 40 years ago. We specialize in design and producing high-shear fluid processors using our core induction chamber technology, which I will explain in a minute. We're local here, actually, in Massachusetts. We're just outside of Boston, about 30 minutes from here. But we have a global presence with customers all over the world. As I mentioned already, our technology serves many industries, primarily pharmaceutical and biotechnology industries. And we're used in both R&D and commercial manufacturing for many drug products and vaccines. But our technology is also widely used in several other industries. We have extensive experience in applications and machine designs, especially around CGMP, which allow us to offer tailored solutions to our customers. So the technology is pretty straightforward, as illustrated here in this diagram. Product with large particles enters the system and is pressurized by a constant pressure pumping system, where the pressure can go up to as high as 30,000 psi or 2,000 bars. And then that pressure, the product is passed through a fixed geometry induction chamber, where material receives consistent shear rates and impact forces. So all material receives exact treatment to achieve consistent and uniform results. Product temperature can be efficiently controlled afterwards if needed. We offer two different induction chamber designs. So this shows the internal structure of the induction chamber. On the top here shows the white type chamber. So the white chamber has two microchannels and product impinges on each other afterwards, which makes them ideal for producing most drug delivery nanoparticles, including LNPs, liposomes, nanoemulsions, and polymer nanoparticles. the one down below here is the Z-type chamber. The Z chamber only has one microchannel and is recommended for processing samples as solid, including doing cell disruptions. Before I move on to show you some results, I'd like to quickly explain how we scale up with our guaranteed scalability. So unlike some other technologies, when we scale up, we don't increase the size of our microchannel, so we don't give you a big channel. Instead, we use multiple parallel aligned channels with identical size. So as long as you keep all other processing parameters the same, material will receive identical treatment inside each microchannel. That's achieve linear scalability. Now with that, I want to discuss a little bit more about lipid nanoparticles or lipid-based nanoparticles. So I think a lot of you knows there's a common method to produce LNPs with the solvent and anti-solvent method. As shown here, there's a few different ways that how you can mix the solvent or anti-solvent streams. But regardless of the mixing method, this is basically a bottom-up method where you generate individual nanoparticles while controlled precipitation. So as I mentioned, when using our microfluidics technology, we're essentially a top-down method. As I mentioned before, we start with large particles and reduce them down to the desired size. And with the system, you can either choose similarly one-step in situ encapsulation, which means you mix your lipid ingredients and your nucleic acid, and you process through, and you achieve in situ encapsulation of the nucleic acid. Or better yet, you can try a different approach, which is a two-step approach. So in the two-step approach, you actually create empty or blank nanoparticles first, and you load your nucleic acids afterwards, typically via complexation. So if you do in situ encapsulation, it is believed, it is considered that the nucleic acids are encapsulated inside the nanoparticles, and if you do the two-step approach via complexation, generally people think that the nucleic acids are absorbed onto the surface. Some people also call this system as lipoplexis. So it seems to be advantageous that you have the RNA or nucleic acid encapsulated inside that's being protected by the particle, right? There's a couple of things. So the system may not be stable, and the exact morphology of these particles are still under debate. This is an ideal situation. And some recent studies also showed that this system can do just as well as compared to internal encapsulation, obviously with some additional benefits, which I will explain you in a minute. Before I talk about that, I want to just quickly show you a proof of concept study we did. Here we processed two different LNP formulations, that's shown here, which one mimics the Moderna's COVID vaccine formulation, and the other mimics Pfizer's formulation. Please note here, both formulations were supplied as a pre-measured kit, so during the process, we did not add or introduce additional solvent. Also, as a quick proof of concept test, we only produced the empty lipid nanoparticles. And also, the entire process hasn't been super optimized, but you can already see still the resulting nanoparticles has good size, with average size just a little over 100 nanometers, and PDIs are around or below 0.2. Now back to the morphology of the LNP RNA structures. So we want to obtain a nice spherical particles, but as indicated by this study, at least you can get different structures, as example shown here, which is a non-spherical blebbed particles. So even without adding any dye to the system, you can already see that this is the blebbed compartment, shows distinctive multiple mass density, which was later on confirmed as resides with RNA. If you can see, I'm not sure, I hope you can see better here as pointed, but this blue arrow here, that this blebbed compartment, when the dye, RNA binding dye was introduced, it also serves as the contrasting agent. you can clearly tell that this bleb is filled with RNA, and this is actually the main lipid body. So this indicates that the RNase has dissociated with the lipids in this case. However, if you do get the spherical particles, it shows that the mRNA was associated with the lipid and has been fully encapsulated inside. And the two stage or morphology may be interchangeable, as indicating here by less prominently blabbed nanoparticles. The study also investigated the physical degradations of this, the LNP RNA systems. When the system is under stress, here just simply underwent freeze thaw conditions. So you can see both particle aggregations, as this one here, or disruption can occur. So when the particle bursts, it can either form smaller, as indicated by this red arrow here, a smaller liposomal structure with RNA encapsulated, or liberated RNA, as indicated by the blue arrow here. Obviously, in this case, you lose the encapsulation efficiency here. And the physical degradations are also reflected in the particle size distribution profile, as you can see the formation of the larger particles. So it's very interesting. The study provides some great insight into the structure or morphology of the system. It demonstrated that obviously the mRNA, remember from the blabbed particles, and it can dissociate from the charged lipid and reside in that blab, which is actually filled with solvent as well, and certainly in a different chemical microenvironment. So, and also it can potentially go through certain degradations that lost the RNA encapsulation. So there's more study needs to be done. The exact effect on this morphology, different morphologies and degradations to the efficacy is still not well understood, but certainly I think this is important, needs to be considered. It is certainly related to both formulations and preparation method. So if you're working with these particles and for developing drug products, you certainly need to consider this side. Now, let's continue to look at the system. As I mentioned, that internal encapsulation seems very good, and but if how is it going to be if we compare exterior complexation with in situ encapsulation? So it's partially answered in this study here. So here, 3 different formulations were prepared. One with an ionizable lipid, which is the C12-200. The other 2 with cationic lipids, DDE and DOTAP, to formulate 3 different kinds of lipid nanoparticles that encapsulating self-amplifying RNAs. So the saRNA are either loaded interior with encapsulation or exterior to the surface by complexation. So the particle size data shown here indicate that the position of the saRNA, whether it's the interior or exterior, or the types of lipids, has limited effect on particle size and distribution because no significant differences were observed. Also, most systems still displayed positive surface charge, only except for this C12-200 with exterior formulation, where negative Z potential was detected. So this indicates that the lipids may have been saturated due to RNA absorption. Also, from the morphology, there's no significant differences between the different formulations. They all displayed circular shape or spherical shape. Now let's look at the results on protecting the RNA and transfection efficiency. So this graph shows here that all formulations will actually offer protection against the direct RNase degradation of the saRNA, with some of the formulations actually achieve close to 100% protection. What's interesting here is with the DDA and RNA absorbed on the surface, actually almost 100% of the RNA was protected in this case. And opposite trends were was observed for DOE type. So despite variations in direct RNase degradation protection, the in vivo transfection efficiency also showed high level efficiency. I want to point out here that with both cationic lipid nanoparticle formulation with siRNA on the exterior, actually offered the highest amount of transfection efficiency and comparable to the ionizable lipid. And again, continue the investigation with in vivo protein expression. Good levels of expression have been achieved. And you can see that, again, the DDA with siRNA absorbed on the surface achieved the highest level of expression, and same as the ionizable lipid. So the last graph shows here is the immunization study with the model antigen. So you can see the antibody responses are the same story. It's pretty high. What's interesting here is with again, with the both cationic lipids and RNA absorbed on the surface, actually achieved the peak level only just after the first injection, whereas the other formulation has to slowly coming up and reach to the peak after the second dose. So with the good indication or results, this I showed you earlier by comparing the exterior complexation. Now let's look further with a real-life example here. So this is a platform developed by HDT Bio using a proprietary lipid inorganic nanoparticle formulation, or LION nanoparticles, for delivering self-amplifying replication RNA. So the results shown here is a formulation developed for coronavirus disease. Again, they use the two-step approach I mentioned, which means the empty or blank lines particles were created first, in this case with a microfluidizer processor. After creating the particles, the RNA was simply introduced via complexation. And you can see from the particle size here, the blank particles showed a uniform small particle size around 50 nanometers, and after loading the RNA, the particle size grew slightly, which also an indication of successful complexation. As expected, the blank nanoparticles were very stable, at least in this case up to 12 weeks, even under the room temperature condition. Even the LNP RNA complex showed good stability, as shown here, that the particle size stayed constant for up to 14 days or 2 weeks at both room temperature condition or refrigerated condition, with the RNA remained its full integrity, as shown here. This slide shows some animal immunization studies. So the graphs on the left shows the mice immunization results. So you can see here that good antibody responses were achieved with different dose groups, especially for the 10 and 1 micrograms dose groups, even after a single injection. So all dose groups achieved good antibody responses after a booster dose. The formulation primarily induced the Th1 biased immune responses, as showed by higher IgG2 titers. The bottom graph here shows the good T cell responses has been detected in both spleens and lungs. So follow the good results of the mice study, they move on to non-human primate immunization studies. So here macaques were injected either with a single dose of 250 micrograms or 50 micrograms primary dose followed with the booster dose. You can see the high dose group developed antibody responses quicker than the lower dose group, but the IgG concentration continued to increase for both those groups and eventually reached to the same plateau. All macaques.
Check 1 minute.
Oh, okay. All those groups showed good T cell responses against SARS-CoV-2 S proteins, as well as showed similar neutralizing titers against the wild type when compared to samples from convalescent humans. So the good preclinical study shown here lead to the continued development of this platform and the formulation. It has since gone through clinical trials and have been actually eventually approved in India under the commercial name of Gemcovec. So that is actually the first ever approved siRNA based vaccine for human use. So the success story of this platform and its great potential of this platform lead to right now they're using this similar platform in many other vaccine and cancer treatment development. Very recent study they just published. They used a similar formulation and successfully prevent mother-to-child transmission for both Zika viruses and HIV viruses in rabbits. So with that, this is my second last slide. Just want to quickly show you our diverse product lines. And the top shows our several different models of lab machines, and the bottom shows our pilot and production systems. With all the pilot and production system and this one lab model, we also offer a pharma version, or which is capable for cGMP processes. And with that, I'd like to thank you for your attention, and happy to answer any questions you have.